Mitochondria play a vital role in maintaining cellular function. While Delta's initial work focused on in vitro measurements of mitochondrial function, such as cell cultures, its later work focused on identifying the role of in vivo aspects of mitochondrial function, or what happens inside the human body in response to BX. Measuring mitochondrial function within the living environment is a field that has, for the most part, remained undeveloped. Current methods have focused largely on in vitro measurements, particularly of the electron transport chain, as previously discussed. However, the need to measure mitochondrial function within the body is critical, especially when one desires to measure the effects of treatment. Accordingly, Delta has developed several practical methods for measuring mitochondrial function in vivo, including open respirometry, crenation studies, mitochondrial function profiles. Open respirometry is often used in athletic performance testing. This technique provides information on the metabolism in general, such as rates of carbon dioxide production, VCO2, and oxygen consumption, VO2, but can also provide additional information concerning mitochondrial function indirectly. For instance, several studies have indicated a significant correlation between VO2 max and mitochondria. VO2 max is a measurement of how well the cardiorespiratory system and muscles are using oxygen. The VO2 score is a measure of the lungs' ability to process the air, the heart's ability to pump the blood's ability to transport the oxygen, and the capillary's capacity to deliver oxygen to the muscle, all functional measures of the body's oxygen delivery system. However, several studies have indicated that the muscle's ability to extract the oxygen is dependent on the mitochondrial function within those cells. Other studies have shown a relationship between VO2 max and several chronic pathologies, including cancer. Delta has demonstrated that an improvement in the function of mitochondria corresponds with an improvement in oxygen consumption in the tissues, hence an improvement in VO2 max. 56 patients suffering from a variety of chronic pathologies including cancer, cardiovascular disease, and various autoimmune conditions underwent respirometry testing before and after BX administration over a 30-day period. Using VO2 max and VO2-CO2 exchange ratios, alterations in skeletal muscle respiratory capacity demonstrated a 33% improvement, corresponding with an improvement in specific diagnostic markers related to the given condition at approximately the same rate, 28 to 37%, demonstrating that general improvement in oxygen consumption within the tissues corresponded to improvements related to the condition itself. Another strategy helpful in obtaining qualitative measures of mitochondrial function is a test known as EPT, or erythrocyte permeability test. The test measures the osmotic pressure of normal red blood cells as measured against disease cells. Healthy, intact erythrocytes become crenated or shrivel up in the presence of a 1% sodium chloride solution. They are able to shrivel up because the proteins in the cell membrane enjoy ample ATP energy to perform this specialized function. However, diseased red blood cells subjected to the same test solution responded by swelling, due largely to the abnormal osmotic pressure inside of the red blood cell. Since approximately 40% of the ATP that a cell generates is consumed by the cell wall integrity could be seen as a qualitative measure of energy production in the mitochondria within tissue-specific counterparts. In 2013, Delta initiated a clinical study aimed at testing the effects of BX on erythrocyte crenation. Using an advanced microscopy evaluation technique, blood was collected from 37 patients suffering from a variety of chronic pathologies. The blood was mixed with a 1% solution of sodium chloride, placed on a slide with cover slip, and recorded using a digital camera for 60 seconds. This procedure was repeated at regular intervals for 30 days. The average baseline crenation numbers, health function, measured prior to the study was 67%. At the conclusion of the study, the average crenation percentage rose to 98%, demonstrating a return of cell wall permeability brought about by increased energy production, and once again corresponding with an improvement in specific diagnostic markers related to the given condition. Another test useful for assessing improvements in mitochondrial function within the body is the mitochondrial function profile, developed by Acumen Laboratory in the United Kingdom. This blood test combines several tests intended to assess mitochondrial function in specific areas, providing specific measures of ATP production within the mitochondria, ATP profiles, NAD functional B3, SODase, L-carnitine, cell-free DNA, glutathione peroxidase. Delta has developed a scoring algorithm that employs Pearson's correlation coefficient and generates a score that combines open respirometry, crenation, and the mitochondrial function profile in order to achieve a more complete picture of the patient's progress. In a 6-month study, including 11 patients, results demonstrated a mean mitochondrial functional score improvement of 39% as compared with the baseline scores. Overall ATP profiles indicated a 27% improvement overall, with 9 of the 11 participants returning to normal ranges. Delta's clinical strategies provide an effective measure of mitochondrial function within the living environment. This broad correlative framework enhances Delta's ability to quantify functional improvements that span across the full biological terrain. From the root cause to the macrobiological systems at large, the BX protocol is designed to deliver complete and comprehensive health restoration.